Physiology of excitable tissues. Nerve tissue. Principal cell types that make up the nerve system. Neurons, specialized for generation of AP, sensory function, generation of thoughts, storage of memory, integrates idea, coordinates muscular activities, compares with other cell type, neurologia, aka neurological cell, non-excitable cell, more numerous, can multiply after maturation, potential cause of glioma, brain tumor. The neuroglia, found in association with neurons, provide supporting function to the nervous system. Microglia, small cell less numerous than astrocytes, act as the macrophage. Microglia can migrate, do not originate from neural progenitor cell. CNS, housekeeping cell. Oligodendrocytes produce the myelin sheath plus metabolic support. 1:50 axons, the last cell type to be generated. Astrocytes, star-shaped, abundant, highly branched, 1:20 million. Fibrous, few long processes, typical in white matter. Protoplasmic, many shorter branched processes, predominate in the gray matter. The larger processes of all astrocytes are reinforced with bundles of intermediate filaments made of glial fibrillary acid protein GFAP, which serves as a unique marker for astrocytes, most common source of brain tumors. Function: form is the blood-brain barrier and thereby regulate the entry of substance from blood into brain tissue, interconnect nerve fibers and blood vessels, provide nourishment, support the network in brain and the spinal cord, maintain the chemical environment of ESF around CNS, provide calcium 2 plus and keep potassium plus and regulate neurotransmitter level in synapse. Regulate recycling of neurotransmitter during synaptic transmission. Neuroglia, ependymal cell, columnar or cuboidal cell, inline the ventricles and the central canal of the spinal cord. Secrete CSF and assist in its circulation. Serves as reservoir for neuro regeneration. Schwann cells, neurolemnocyte, form myelin sheaths, vital to neural regeneration. Satellite cells, small cells that line the exterior surface of the PNS, regulate the external chemical environment, are very sensitive to injury and may exacerbate pathological pain. Neurons, different from other cells by two ways, has axon and dendrites. Neuron does not have centrosome. Neuron has three distinct parts. These are cell body, dendrites, and axon. Dendrite and the cell body, input zone. Nucleus, axon, hillock, trigger zone, axon, conducting zone. Soma contains nucleus plus most normal organelles. Biosynthetic center of the neuron contains a very active and developed rough endoplasmic reticulum rare, which is responsible for the synthesis of NTS. The neuronal rare is referred to as the Nisa body, contain many bundles of protein filaments, neurofibrils, which help maintain the shape, structure, and integrity of the cell. Soma. In the soma above, notice the small black circle. It is the nucleus, the site of ribosome synthesis. The right circular area around it is the nucleus. The mottled dark areas found throughout the cytoplasm are the nasal substance. Nasal bodies and neurofibrils are found only in nerve cell and not in other cells. Dendrites are distinguished from axons by the presence of nasal granules under microscope contain multiple mitochondria, act as a receptive surface for interaction with other neurons. Most somata are found in the bony environs of the central nervous system. Cluster of somata in the central nervous system, nuclei. Cluster of somata in the peripheral nervous system, ganglia. Neuronal process, arm-like extension emanating from every neuron. The central nervous system consists of both somata and processes, whereas the bulk of the PNS consists of processes. Tracts, bundle of processes in the CNS. Nervous, bundle of processes in the PNS. Two types of processes that differ in structure and function, dendrites and axons. Dendrites are thin, branched processes which main function is to receive incoming signals. They effectively increase the surface area of a neuron to increase its ability to communicate with other neurons. Small mushrooms, shaped dendritic spines, further increase the SA, convey towards the soma through the use of graded potential, which are some that similar to action potential. Neuronal process, axon. The site where two neurons or a neuron and an effector cell can communicate is terminus synapse. The tip of most axon terminals swell into synaptic bulbs. These bulb-shaped structures contain synaptic vesicles, tiny sacs that store chemicals called NATs. The neurotransmitter molecules released from synaptic vesicles are the means of communication at a synapse. Neuronal process, axonema, axon plasma membrane surrounded by a myelin sheath, wrapping of lipid which protects the axon and electrically isolates it. Increase the rate of AP transmission. This wrapping is never complete. Interspersed along the axon are gap where there is no myelin. These are nodes of Ranvier. In the PNS, the exterior of the Schwann cell surrounding an axon is the neurolemma. Type of neurons, morphologically classification of neurons, multipolar neurons, several dendrites and one axon. Most neurons in the CNS are motor in function, bipolar neurons, one main dendrite and one axon. In the retina, the eye, in the inner ear, and in the olfactory area, unipolar neurons have dendrites and one axon that are fused together to form a continuous process that emerges from the cell body, sensory in function. Anazonic neurons with many dendrites but no true axon do not produce action potential but regulate electrical changes of adjacent neurons. Functional classification of neurons. There are three classes of neurons: sensory or afferent neurons. Most sensory neurons are unipolar in structure, conduct impulses from periphery to the center. Motor or efferent neurons, interneuron, association neurons. 90% of the neurons in the human central nervous system, multipolar, anazonic type. Adrenal fibers. They are myelinated fibers, function motor to the skeletal nerve muscle, diameter 10 to 20 micrometer, conduction, velocity 60 to 120 ms per second. Type A fibers, myelinated fibers, motor function, diameter 5 to 10 microM, conduction velocity 30 to 60 mPs. Type AO and AY fibers, myelinated, motor function, diameter 1 to 6 microM, conduction velocity 6 to 30 mPs. Type B fibers, preganglionic autonomic fibers in the myelinated, diameter 2 to 6 microM, conduction velocity 60 to 30 mPs. Type C fibers, post-ganglionic fibers, unmyelinated, diameter 0.5 to 2 micrometer, conduction velocity 0.5 to 2 mPs. Excitability of neural tissue. What does excitability cell mean? Excitable cell, a cell which has a potential of producing an electrical signal in response to the stimulus. Membrane potential. Definition: electrical energy difference between the inside and the outside of the cell. EM equals to VM minus V out, where VM potential on the inside of the cell, V out potential on the outside. All cells have membrane potential. EM equals to membrane potential. The range of EA equals to minus 20 MeV to 90 minus 90 MeV, and any range change of membrane's permeability of ions causes a change in EM. Resting membrane potential, RMP, the membrane potential at rest, no net flow of ions across the plasma membrane, always negative in nerve and the muscle cell. Magnitude is K for individual cell type. RMP is necessary for the cell to fire an action potential. The RMP of a typical neuron is minus 90 MeV. What are the causes of the RMP? 1. An outward diffusion of K positive through K positive leak channel. The sodium positive minus K positive ATPase. 3. There are negatively changed non-diffusible proteins. Basic electrophysiological terms. Excitability, excitable cell, stimulus, threshold stimulus, polarization, depolarization, repolarization, hyperpolarization. Role of ion channel. Changes in membrane potential are due to changes in ion movement across the membrane. Movement is through ion channel. 1. Leak channel. 2. Gated channel. 3. Voltage gated channel. 1. Voltage gated channel. 2. Chemically gated channel. 3. Mechanically gated channel. Classification of ion channel. Leak channel, gated channel, sodium plus channel. There are 3 major types: sodium plus leak channel, voltage gated sodium plus channel, ligand or chemical gated sodium channel, K channel. There are 4 major type classes: K positive leak channel, voltage gated K positive channel, 3 ligand gated K positive channel, 4 G protein gated K positive channel. Calcium plus channels are the following: voltage gated calcium positive channel with subtypes: L type C plus gated channel, T type C plus gated channel, N type C plus gated channel, ligand gated C plus gated channel, G protein gated C plus gated channel. Action potential, nerve impulse. Action potential is a rapid, conductive, and irreversible change of the membrane potential after the cell is stimulated. 65 MeV is said to be the threshold for stimulation. Features: all or none phenomenon, always be full-sized. It will not get lost along the way. Rapid and reliable information. Has threshold, amplitude, and duration is K. Always depolarizing. Has refractory period. Non-discriminal. Propagation is active. Cannot be summated by membrane depolarization. Action potential: if VM reaches threshold, N channel open and N positive influx depolarizing the cell and causing the VM to increase. This is the raising the phase of an AP. Eventually, the N plus channel will have inactivated and the K channel will be open. Now, K effluxes and repolarization occurs. This is the failing phase. The K channel are slow to open and slow to close. This causes the VMEs become hyperpolarization. Voltage gated N channels. Two gates, activation gates, voltage dependent, open with depolarization, fast opening, positive feedback. Inactivation gates, voltage dependent, closes with depolarization, open with hyperpolarization, time dependent, slow. Voltage gated K channel, one gate, voltage dependent, sensitive to depolarization, time dependent, open more slowly than sodium channel, negative feedback, slow closing result in hyperpolarization. Refractory period of decreased exactability following an action, potential, absolute refractory period, importance, limits the rate of firing of AP, prevents AP from traveling in the wrong direction along the axon, relative refractory period. If an AP is generated at the axon E-lock, it will travel all the way down to the synaptic lobe. Continuous conduction, saltatory conduction. Continuous sweeping conduction occur in unmyelated axon. The whole length of the membrane is depolarized. Velocity of conduction is low. Consume large amount of energy. Saltatory or jumping conduction occur in myelinated axons. AP occurs at the axon of nodes of Ranvier. Velocity of conduction is faster, consumes few amount of energy, economizes ATP. Orthodromic, vice versa, antidromic conduction. Orthodromic conduction, soma axon thermal, antidromic conduction, axon thermal soma. Saltatory conduction, node of Ranvier. Only the region where current flow across the membrane exists, a region of concentrated voltage gated sodium channel. Factor affecting rate of AP conduction, presence or absence of myelinated, diameter of fiber or size of nerve fiber, age, temperature. Synapse, the region where there is a transfer of message from a neuron to the next. The junction between the two cells in the one must be a neuron. There are three types of synapse: neuronal junction, neuromuscular junction, neurogranular junction. There are three types of neuronal junction: axodendritic junction, axosomatic junction, axonic junction. Component of axosomatic synapse. Presynaptic terminal contains neurotransmitter NT. Synaptic cleft contains ECF and enzymes. Postsynaptic neuron contains receptor for the action of NT. Mechanism of chemical synaptic transmission. An AP enriches the presynaptic axon terminal of the presynaptic cell and causes V-gated calcium plus 2 channels to open. Calcium over 2 plus rushes in binds to regulatory protein and initiate NT release by exocytosis. NTS diffuse across the synaptic cleft and then bind to synaptic receptors on the postsynaptic membrane and initiate postsynaptic potential. NT receptor interaction result in either EPSP or IPSP. Mechanism of chemical synaptic transmission. When the NTR combination triggers the opening of ligand getting N channel, this leads to membrane depolarization, EPSP. Example, AK on nicotinic receptor. When the NTR combination triggers the opening of ligand get K or CI channel, this leads to membrane hyperpolarization, IPSP. Example, GABA or GAP on GABA B receptor. Neurotransmitter removal. NTS are removed from the synaptic cleft via enzymatic degradation, diffusion, reuptake. Synaptic transmission. Two modes of transmission: electrical transmission, chemical transmission. Advantage over electrical synapses: one way diffuse direction of communication. Presynaptic cell to postsynaptic cell allows two different kind of signaling event. Sequence of events during synaptic transmission. One, on the base of function synapses and are divided into two types: excitatory synapses, which transmit impulses. EPSP has two properties. It is non-propagated. It does not obey all or one flow. Two, inhibitory synapses, which inhibit the transmission of impulses. Inhibitory function. Excitatory vice inhibitory synapse. Excitatory, more likely to have action potential, depolarization. Inhibitory, less likely to have action potential, hyperpolarization, membrane stabilization. Excitatory synapse, depolarize postsynaptic cell, brings membrane potential closer to threshold by opening or closing ion channel, opens channel that are equally permeable to N and sodium and K, causes depolarization because of the stronger force of sodium to flow into the cell. Depolarization, EPSP confined only to the synapse. It is a graded potential. Inhibitory synapse, neurotransmitter bind to the receptor channel for either K or CI open, hyperpolarize the cell. If K channels open, K moves out. IPSP, inhibitory postsynaptic potential. If CI channel opens, either CI moves in. IPSP, CI stabilizes membrane potential.